So this is the quick 5-minute quiz 2.6 review. This week we've been talking about the two kinds of nucleic acids, DNA, which we have here on the right side, this double-stranded guy, this double helix, that's DNA, and the single-stranded cousin, RNA. So remember that nucleic acids are both polymers of something called nucleotides. So polymer just means a chain, and in this case, the student has this really awesome graphic here. So the polymer is the chain of these nucleotides. So this whole thing would be a polymer. This is one strand of DNA, one polymer of a strand of DNA. And she has here the nucleotides. So these would be the monomers of DNA and RNA. They're both nucleotides. Okay, so DNA are polymers of nucleotides. So let's take a closer look at this nucleotide structure that you guys have been drawing. So remember that this is your phosphate. That's where we're depicting as a sugar that's connected to this pentose sugar. So they're different between DNA and RNA, and that's connected to your base here. So phosphate, sugar, pentose sugar, and base. So that's your nucleotide structure. Those are the monomers. Okay, and there are some shared characteristics between DNA and RNA. So each of these nucleic acids has 4 different bases. In the case of DNA, it's A's, G's, C's, and T's. And in the case of RNA, it's A's, G's, C's, and U's. So both of them have A, G, C in common, and both of them, again, are polymers of nucleotides. But there are some differences between DNA and RNA. So the first thing that you guys probably notice is that DNA has 2 strands. It's got this double helix, this double strand, whereas RNA is single-stranded. The other difference that you probably already noticed is over here in the bases, that in DNA, they have a thymine, they have a T, whereas in RNA, they have a uracil. So this is usually depicted as a U. And again, the other big difference between DNA and RNA has to do with the sugars, and it's sort of in the name. So DNA takes its name from the sugar deoxyribose. So see, deoxyribose is going to show up, so it matches D, DNA. So it's actually deoxyribonucleic acid. And whereas RNA has a ribose sugar. And again, that matches with the start of the RNA name. So RNA goes with ribose. And really the only difference between them is that the deoxyribose has just a hydrogen, where ribose has hydroxyl groups on both these. So know what's in common, that they are both polymers of nucleotides, that they both have these A's, G's, C's, and A's, G's, and C's, but know what sets them apart. So, and that's in both their structure, in these bases here, and also in the sugar structure underneath them. Okay. So again, DNA is made up of these 2 polymers, these 2 polymer strands of nucleotides that fold into that double helix, this model you're probably familiar with. And remember that the strands of DNA, when they're paired, they are anti-parallel. So this one, again, remember the sugar, you know, it looks like an arrow pointing up. If I look at the base pair across way, it's flipped upside down. So this sugar is flipped upside down compared to this one. This whole nucleotide is flipped upside down compared to this one. So where one strand is upright, the strand on the opposite side is anti-parallel and pointing downwards. So when you guys are drawing these DNA molecules, I would like for you to have these sugars pointed down to depict that anti-parallel direction. Okay, because these nucleotides are flipped on this side compared to this side. One is pointing up, one is pointing down. Okay. So again, be able to identify, you know, where your phosphates are. If I'm asking you to draw DNA, I would like you to tell me which sugar it is. Don't just say the sugar. This is deoxyribose if we're in DNA. It's ribose if we're in RNA. And again, these are your bases here. And be able to identify those hydrogen bonds as well as the covalent bonds. So hydrogen bonds are going to be what's in between here, holding these bases together. And the covalent bonds are what's, you know, really linking this sugars and phosphates together. Okay, and again, remember your DNA base pairing rules. A's always pair with T's, G's always pair with C's, and again, it's these hydrogen bonds that are forming between the base pairs. You might see them as straight lines sometimes, but it won't matter. If they're in between these bases, they're hydrogen bonds. Okay, and that's all you guys need to know for quiz 2.6.